It's late July, and photographer Jim Hellam has returned to the Caribbean Sea, to an isolated island 75 miles south of Cuba called Little Cayman. There are many features of this underwater paradise that attract inquisitive divers from around the world.
On this reef here are thousands upon millions of books.
Successions. I'm a geologist. I'm looking at the sequence of events that occur over time, and I can see that.
Big enough to see, but too big to photograph.
We're really interested in moment-by-moment thought process.
One wall, rich with questions, rich with life, rich with what marine experts call ocean biodiversity.
Biodiversity is everything that is alive, and a big part of that biodiversity lives and comes from the ocean.
You can think about it in terms of, all of it, the whole thing together makes nature function as it does.
And nobody knows exactly what is it, the equilibrium that is needed on this planet to make it something in which we can live.
These divers each bring their own questions to Little Cayman this month. But they'll also be assisting each other, exploring the many features of Bloody Bay Wall.
This entire island is surrounded by walls. It's really the top of a mountain. It's a pinnacle sticking up from the Cayman Trench.
Just a stone's throw from shore, the cliff walls of the underwater peak plunge down 5,000 feet.
This particular place is known for Bloody Bay Wall. It's how I heard of it. Bloody Bay Wall was purported to be one of the most pristine communities of animals and marine life in the world.
It wasn't always so pristine. Local legend holds that the waters ran red with human blood in the late 1600s from a fierce battle on the surface between British and invading pirates. And on these beaches in the 1700s, well over 2 million green turtles were hunted for food. But in 1986, Bloody Bay became a park, protected from local fishing and reef damage. Ocean biodiversity was given room to flourish. Biodiversity provides the air we breathe, the weather we feel, the fish we eat, and the beauty that inspires Jim Hellam. In 1999, Jim made multiple dives on a section called Great Wall West, recording the highest resolution composite image of a coral reef ever captured. A photo so large that a print would be 70 feet wide. The team is recording a second composite image this month, more than 10 years after the first, and of the exact same section of wall. Together, these two images will reveal how life here has changed. Jim is not expecting to see the kind of dramatic loss of biodiversity found elsewhere in the Caribbean. Places where coral reefs are less protected, where they no longer function as they once did. As Jim descends the Bloody Bay Wall, he benefits from the reef knowledge of his friend Carrie Manfrino.
What kind of turtle did you guys see?
Founder of the Central Caribbean Marine Institute on Little Cayman.
We are interested in sustaining the marine biological diversity in the ocean through research, education, and conservation.
Carrie's team maintains equipment to monitor the reef in real time. They also conduct surveys of coral, sponges, and fish. There's a lot to keep track of. 1 out of 4 marine species in the world's oceans spend some time on a coral reef. even though reefs are covered by only 0.1% of the world's ocean surface. Some of the species have extraordinary capabilities. Two of Carrie's colleagues are working to find species that could solve a biomedical problem in a faraway Yale University laboratory.
And then now, when we this one here, I'm going to move this down 6, right then and there.
My own personal interest in the laboratory is to understand thought process by visualizing neuronal activity on a large scale. So in other words, when the body executes a certain behavior, how the brain assembles that information in the motor system. And we really don't know how that works. And our goal in the human world is, of course, to help people who have lost functionality of their body.
The researchers team up with Jim Hellam, seeking something that Vince suspects will help him visualize a nervous system at work.
I know, but I'm trying to save your life.
Coral fluorescence. Best seen at night. Marine biologist David Gruber.
There is major competition for space. Everyone's fighting for their place on the reef. In doing so, you've got strange, wild adaptations that you don't find anywhere else.
The divers know that one of these adaptations, lurking out there somewhere within certain corals and jellyfish, enables them to emit fluorescent light. Not easy to see, but visible when excited by a certain technique that these divers are using.
We bathe the reef in very pure blue light.
Nobody knows why submarine life fluoresces, but the team knows how. A single piece of DNA is responsible, and that DNA has proven to be extremely valuable to research. In 1994, a scientific paper outlined how it can be copied from sea life and inserted into the cells of many other life forms in the lab. Any cell tagged with that DNA is then visible. With this cellular lighting kit, researchers can see what cells do.
Since that paper came out in 1994, there's been 20,000 papers using this. It's become possibly the most important tool in biomedical research today.
Most of what they are seeing tonight is green fluorescence, fairly common. But other fluorescent colors can also be isolated. So far, about 150 have been found worldwide. Of those, David and Vince have found over 30. But they've not yet found the one that Vince needs. He seeks the long wavelength of a far red fluorescent light. With the DNA that causes this placed within the nerve cells of the brain, Vince can watch and even record brain activity in real time without disturbing the skull. This has never been done.
Gorgeous. It's like a psychedelic light show when you put the blue lights on it. It just glows this gorgeous green, fluorescent green.
After several dives, David and Vince, with the help of Jim Hellam, have collected enough imagery to identify potential new fluorescent proteins. They head home to process their findings. Jim finishes his daylight photography of the wall for his own work and begins piecing together over 400 individual exposures into a composite image of the wall.
Now I have the whole image, 70 feet wide, 20 feet high, and I have it captured in a way that has the true detail and color to the point that we can zoom in and see a 1-inch fish.
Months later, Jim displays a completed photo mosaic of what the reef looks like in daylight. Even on this protected reef, change over 10 years is evident. The wall is greener, less colorful coral, more algae. The green algae indicates that the coral cover of Bloody Bay Wall and Little Cayman is under stress and shrinking. Carrie Manfrino's surveys on 8 sites have indicated a reduction in coral cover from 28% to 16% since 1999. One known cause is coral bleaching, a result of the warming seas observed around the world. Local reef conservation will not protect against global warming. Another change is the arrival of the voracious lionfish. With no predator to check its numbers, it ranges, free to feed on many local fish, threatening to reduce biodiversity. Carrie's team will be closely monitoring lionfish in the park. Jim has also processed an overlay, mapping what the corals on Bloody Bay Wall look like at night. Spectacular displays of green fluorescence. But the far red light that Vince and David seek has not yet been seen in any marine life on Bloody Bay Wall. They will continue that quest here and in other oceans. Successfully finding it will not only help with Vince's brain research, but also the research of others working with AIDS and cancer. Fluorescent protein is only one quality of one life form in a living ocean with thousands of different species and capabilities, yet to be explored.
We do not know today which are the ones which are really so vital that our own species is going to be protected in the future.
This is a purely selfish reasoning to study this. Like, we don't need another reason to help protect coral reefs, because they're beautiful places and they are biodiverse and everything. We didn't need another reason, but there is strong reasoning to think that dramatically, as an organism disappears like this on the Earth, that enormous history of evolution disappears instantly with it.
We might be able to break some of it down and still be okay, but eventually, if you destroy too much of it, then the damage may be irreversible, or you will lose a lot of the functions that we're benefiting from right now.
Dave and I view it kind of as a library of genetic material.
And we're just burning the books and we're throwing them away. Never got a chance to read them.
And the problem is the loss of biodiversity. That's when you cannot do anything. When you have lost a species, it's lost. Finish.
In this complex, dense environment, we just have to dip in there and look at it as long as it's still there.